Okay. First, thanks to Aaron and Mercy and the department gave me this opportunity to talk about the work from our lab. And my name is Kai, and I'm a second-year postdoc here in Maureen Sauter's lab. And originally, I'm from China, and I've been US since 2014, and I got my PhD from Miami University, Oxford, Ohio. And I moved to Michigan early 2019. And my research work is mainly focusing on, it's in a collaboration with Dr. Dina Dolinoy, who is a professor in School of Public Health. My research work is mainly working on the Target 2 consortia, which is related to the epigenetically modification of environmental exposures. And today, the topic I'm giving is work that is published, I think it's early this year. And I just use a title from the paper that we published. And the title is Testing Proximity of Genomic Regions to Transcription Start Sites and Enhancers Complements Gene Set Enrichment Testing. And this paper is published on Frontier in Genetics. And okay, so the background here is gene set enrichment methods are commonly developed to determine the pathway associated with genome-wide sequencing data, like ChIP-seq data and ATAC-seq data. And but most GSE methods were developed for RNA-seq data, which is not adjusted for the various length of genes or regulatory space between them. And they are not like developed for GSE testing with large set of peaks. And there are some methods that are developed for do the GSE testing of peaks, but they are mainly focusing on the relationship between the peaks and the TSS. But in reality, not all the peaks are like really related to a TSS. In here, because we use a lot of ChIP-seq data, so I just stated transcriptional factors here. So not all the transcriptional factors tend to bind to the TSS. Some of them tend to bind to enhancers. So the question is that, so whether a TF is binding to the enriched gene set, it's whether the TF is because the TF is binding to closer to a TSS or an enhancer or both or either of them. And to our knowledge, there's no method like developed to answer this question. So that's why we developed this method called Proxyrent. It stands for proximity regulation. And the method is like a kind of straightforward. So there are like three steps. So the first step is we measure the distance between the peaks and the closest TSS or the enhancers. And then we normalize the distance based on the gene locus length for the distance between peaks to the TSS and the average distance to enhancer to the distance between peaks to the enhancers. And then we test whether the peaks in a gene site are significantly closer to a TSS or enhancer than expected by chance. And we use Wilson-Coxon rank sum test. So for the materials and methods, so the data set we use here, we downloaded 19 human ChIP-seq data set from ENCODE. And there are 35 TFs in those 90 ChIP-seq data set over 3 cell lines. And the gene set we used is from the go.db bioconductor package. And we only use the biological process go terms. And we not use all of them. We only use gene set with larger than 50 and smaller than 2,000 genes of the go BP terms. Because if a gene set it's like a very small gene set, we'll have like a very little statistical power. And if the gene set is too large, it will be hard to have meaningful biological interpretation. So that's why we only choose the gene set with those numbers of genes. And in total, they will have 500 and 185 GoBP gene sets. And so for the first step, it's measuring the peak distance to the nearest TSS or enhancer. So for here, so the regulatory proximity was defined the distance in base pair between the peak's midpoint to the closest TSS or the midpoint of the closest enhancer region. And the TSS location we used are from those two bioconductor packages. And the enhancer regions are defined by the union of the DHS sites and also processed by ENCODE. And so the distal and the non-promoter elements were defined as those like a 5 kb away from the TSS, like the figure shows here. So so in here we call it enhancer, but it's not only it also like include other elements like silencers and insulators, but we all call them enhancers. Like all the distal and the non-promoter elements, like in this region, we will use them as like an enhancer in here. So we will measure the peak to the distance of those regions and the peak to the corresponding TSS to test whether the related gene of these peaks is like the gene set is enriched. It's like a more closer to the enhancer or to the TSS. And so for measuring the distance between peaks to the enhancers or TSS, so first step is we need to assign the peaks to genes to do the gene set enrichment test. So first is we need to measure the distance, and then we need to like assign the peaks to the genes, then do the test. So first is like because for here we want to test whether the gene set is closer to TSS or closer to enhancer. So for the TSS part, we assign the peaks to the near to the genes by the locus definition is like a nearest TSS. So this locus definition is from the chip enrich package. It's a paper that our lab previously published. So in here, it's like the gene locus definition is something like in here. So those points here, it's the midpoint between two adjacent TSS. So in that way, for this blue gene in this figure here, those two peaks will be assigned to the blue genes because they are like located in the gene locus, in this gene locus. And these peaks is like located within the yellow gene, so will be assigned to the yellow gene. And yeah, so here is a definition of the gene locus length. So the length of the region on the genome is such that a peak of binding in the region is assigned to that target gene, which means, for example, so for this yellow gene, so the gene locus length is like the length of this yellow bar here. And for measuring the distance between the peaks to the nearest TSS, we found out that as the genes with a longer locus length, the peaks will be like far away from the gene's TSS. So that's why we need to do the normalization for the gene locus length. So we found that the gene locus length is a potential confounding variables to this problem. So how do we do the normalization? It's something like this. So this figure is like a example here. So we will like normalize the distance of the peak distance to the TSS like in this way. So first we will combine all the 19 ChIP-seq data together, and then we fitted a cubic smoothing spline like something like this. And so in this figure, the x-axis is gene locus length, and the y-axis is the log distance to the TSS. So the fitted line here will provide the expected global average binding distance for each gene. So the adjusted distance to TSS equals to the distance of the original distance between the TSS minus by the log of the values from this line here. So in that way, after normalization, peaks that are closer to a TSS than expected based on this line, on the value on this line, will contribute to a significant promoter proximity for a gene site. And similar to the normalization for the distance to the TSS, we also normalize the distance to the enhancers, but it's kind of a little different from the TSS. So for the enhancers, also it's if the enhancers it's they are far away from each other. So in that way, so the peaks, the distance of the peaks will far away from the enhancers. But another thing is this distance is also related to the distribution of enhancers of each locus definition, in this figure shows here. So the density of the enhancer per genes is a kind of a confounding variables for this problem. So for example, so when we do these normalizations, what we did is similar to the previous one. So we also fitted a line based on the all 90 ChIP-seq data set. And the x-axis here, it's the average distance to enhancer for each gene. And the y-axis here, it's the distance to the enhancer. So the adjusted distance to enhancer will be the distance to the enhancer, the distance to the nearest enhancer minus by the values from this line here. And for example, when we so the x-axis here is for this peak, it will be assigned to this gene, and within this gene locus, there are 3 enhancers. So we will calculate the average distance of this gene, and the distance to the enhancers. It's will be in the x-axis here, and the y-axis here it's the closest, the distance to the closest enhancers, which is this one here. So then we can adjust the distance by this value minus by this value. And yes, so in that way, so it's we will normalize the distance to the peaks to the enhancers. And after that, for example, if we have a TF, we have ChIP-seq data set here. So we will calculate the distance of all the peaks to the promoters and the enhancers, to the TSS and to the enhancers. And then we will assign those peaks to the genes. And then we will have this. So after that, it will be a normal GSE testing method. So we will have a gene set of interest, and the other gene set will be used as a background gene set. Then the thing that we test is whether the peaks in this gene set are significantly closer to the TSS or enhancer. And in order to make sure that our method is a good way to answer the questions, so we use 3 different ways to test the type 1 error rate. And the first one is we reassigning the every peaks to a random gene. And in the second way, it's we reassigned every peak to a random gene, but all the genes were ranked by the locus length and binned into 100 genes. So it's a randomly gene set. And with those genes are a similar locus length. So it's this is for the distance to the TSS. And also the third way is we reassign each peak to a random gene, but all the genes were ranked by the expected distance to enhancer and bin the genes into a set of 100. So those 3 randomly site was used to test the type 1 error rates. and the results. So for here, it's the workflow for the Proxyrent. So our method is like a complementary approach for a normal GSE test. So in that way, it's like a so the user will like put the input will be like a genome region of sorry. So the input will be the genomic regions of interest. So will be like the chip-seq data results, like a lot of peaks and regions of the genome. And then in parallel, so we will run the Proxyrent, which is in this way here. So it measures the distance to TSS or enhancer. And in parallel, in other ways, like we will assign the peaks to genes to do like a normal gene set enrichment test. Use the same gene set database. And so then we will do the Proxyrent proximity test and the gene set enrichment test, and then combine the result from them together. Because in here, we will test, actually we will test twice. So one for TSS, one for enhancer. And so when we combine those two results together, we will have like a figure like in here. So in here is like here is the promoter result, which is like to test whether the gene set is significantly closer to the promoter, TSS. For the enhancer result, it's like to test whether the gene set is enriched that they are closer to enhancers. And so for here, the y-axis here, it's the result from the proximity test. And the x-axis here, it's a result from the gene set enrichment test. So in that way, we can see that if the gene set located in here, which is like closer to the promoter and also enriched. If it's in here, it's like far away, because this is enhancer result. So if it's located in here, which is that they are far away from the promoter, they are far away from the enhancer and also enriched. So from for the type 1 error rate and so the false positive rates, so we use like the three different non-data set to do this test. And for the p-value like smaller than 0.05 cutoff, we expected the type 1 error rate approximately to 5%. And so here, this one is for the results of to test the distance to the TSS. This is to enhancer. So it looks like it's not like exactly equals to 0.5, but it's very close. And for here also, we choose a p-value smaller than 0.01. So we expected the type 1 error rate is about 0.1%. And it's also those results like very close to the value that we expected. So after that, so we want to see how our method performed on real data set. So we like use our method to process all the 90 chip-seq data set to see how they looks like. And so for here, so this is like the so majority of the 90 chip-seq data set shows a strong negative correlation, which looks like this. So for those two figures, so the x-axis here is the promoter results, and the y-axis here is the enhancer results. So a lot of chip-seq data set that we used in this research, it shows a strong negative correlation, which means that some of them will like very close to promoter, some of them will like very close to enhancer. But a small amount of the chip-seq data set, it looks like this. So it's not like a clear patterns. So then we want to like then we did a hierarchy clustering to see is there any like a different regulatory patterns of those TFs. So when we do this, so here is the results, and here's the results. And how can we do the hierarchy clustering? It's basically we divided those two figures like into different 4 regions. So we just count the numbers of the dots in the 4 regions. So each chip-seq data set will be represented as 8 numbers. And for example, for this one, so this is a promoter results. And so the x-axis here is a GSE p-value. And the y-axis here is a proximity p-value. So if if the dot is like in this way with a larger number on the x-axis, will be like more enriched. If like a larger number on the y-axis, will be like closer to this region. And so when we count those numbers, we just count how many dots was in this 4 regions. And the black dots here are not significant ones, so we didn't count them in. We just count the significant results for both the GSE test and the proximity test. So here is a result of all the 90 ChIP-seq data set hierarchy clustering results. And it showed that there are like 3 different groups. So the first group, it's like the blue groups here. So those TFs, they shows a pattern that they are more tend to be closer to promoters. And the second group is the purple group here. So there's like only 11 of the data set shows in this group. And those groups, they are like more like, those TFs are like more tend to binding closer to enhancers. And the remaining like 32 group data sets is like they didn't show like a clear patterns. So for example, so for this one, this is one data set from the blue group, which is like show a clear pattern that those gene sets are enriched by the TF that are binding close to the promoters. So in the promoter results, there is a clear positive correlations, which is the dots are raising here. They are like enriched and also close to promoter. But in the enhancer, corresponding enhancer results, so they are also like enriched, but they are like far away from enhancers. So in the purple groups, it's like the pattern is looked at this. So they have like clear negative correlations in the promoter results, which is those like a gene set, they are enriched, but they are far away from the promoters. But in the corresponding enhancer results, they are also like enriched, but they are like closer, they are in this region, which means they are closer to the enhancers. And for the third group, so those the results, it looks like those 2 figures. So in the promoter results and the enhancer results, there's no clear pattern to show like a positive correlation or negative correlations. So those are like they're not clear. They didn't show like a clear binding proximities. And
okay, 1 second. This is Marcy. There's a question in the chat, and so I just want to make sure before you get too far along. The question is, how were true positives defined for benchmarking?
Oh, for the true positive, we actually we collect a lot of the ChIP-seq data from, let me remember, sorry. We collect a lot of ChIP-seq data from I think it's from, let me, I think we collected from the bioconductor packages and to do the test. I can show that figure later because that figure is in a supplementary figure. Okay, so for here, after we finish like the gather 3 different patterns, so that we want to identify no association with promoter enhancer bindings to see that, okay, so the result we found is like a real result. So for the promoter bindings here, so we selected this transcription factor. It's called 65 in this cell line. And this TF is involved in the determination and maintenance of the retinal formation. It is also well known to bind to promoter regions of related genes. So the A figure here, it's the distribution of distance from peaks to nearest TSS of this data set. So we can see a clear peak here, so which represent that the distance, those like peaks is like a very closer to the TSS. And here is a promoter results of this data set, and here's the enhancer results. So we can see a lot of genes that they are in here, but in the enhancer results, they like flip over in this region. So which means that those genes that are like more tend to be bound to the TSS and far away from the enhancer. And in details, we selected two like gene set from those figures. So gene set 1 and gene set 2. And in here, so the gene set 1, this graph is the distance to the nearest TSS. So we can see in this gene set, which is like this gene set in here, there's a clear peak that is like in here, so which is like closer to the TSS. And in GSAT2 here, we can see, so in the GSS, it also, because it's located in here, so we can see a peak in here, which is like a far away from the TSS, but clearly the big peak is here. It's also like near the TSS. So in the enhancer results, also it's like the first gene set will be this in here, and this one will be here. And we can see, so for gene set 1, for the distance to a nearest enhancer, which like a clearly peak here, which is like a far away from the enhancer. And for the gene set 2, which is also like a clear peak here, but it also have a small peak here, because in this one they have a small peak here. So this is an example that is known that this TF is like a closer binding to the promoters. And for the enhancer bindings, we selected this transcription factor, which is the NRSF in this cell line. So this TF is known to silence neural genes in non-neural cells and act as a transcriptional repressor or enhancer of target genes. So this transcription factor are often regulated from the enhancer regions. So similar to that one, so the distribution of distance from peaks to nearest TSS, we can see a peak in here, which is like far away from the TSS. And here it's a promoter result, here is an enhancer result. So we can see, we also like selected two gene sets to show the distance to the nearest TSS. So we can see this gene set 1, gene set 2, both of them shows like clear peaks that are far away from the TSS. And in the enhancer result, it's like both of them are like in this region. And we can see there's a clear peak that is in here and here, which is like they are closer to the enhancers. So so after that, so we want to see, so if those TFs, they have like clear patterns in different cell lines. So how are they like how the regulatory patterns in the different cell lines? So we selected this NRSF transcriptional factors to see how the regulation patterns in different cell lines, because in here we have like 3 different cell lines. So in this K562 and the GM cell lines, so it's like this one, all the like significant GO terms identified by both Pauling-Rich and Proxyrent were found like they are closer to enhancers. But in these cell lines, it's like a totally different scenario. So almost all GO terms are closer to promoters. So which is like a very interesting because in different cell lines, the same TF may have performed like a totally different types of the regulatory patterns. So the summary of this research is like we developed this genomic region proximity test called Proxyrent that can be used as a complementary method for the normal GSE test. And it can be used with various types of genomic regions, including ChIP-seq data, TAC-seq data, demethylation, and some others. And it's able to find gene set with region that bind significantly closer or farther from promoters. And this method is available at the chip-enriched bioconductor package. And here's our lab, and thank you. And I will like to answer any questions. Okay, so first, okay, let me answer the first question. For the true positive, okay, for the true positive, what we used, because there's no like a golden standard data set that clearly shows that all the also show that this transcription factor it's like closer to promoter or enhancer. So we used the GoBP terms from the ORGHS bioconductor packages, and we tested. Let me open the supplemental figure. Okay. Okay. Supplemental. Oh yes, so for here, to support the idea of this method, so we compared the percentage of the gene the promoter covered with ChIP-seq in the assigned goal terms and not in the and the peaks that not in the assigned goal PP terms for the TF, and so all of those TFs are from the this bioconductor packages, and that so this figure shows that this figure shows that most of the ChIP-seq data set in this paper, it shows a higher percentage of genes in the TF assigned goal terms with peaks in the promoter region than the genes that are not in the TF assigned goal terms. So basically, we use goal terms from these packages to do to define the benchmarking stuff. This okay. Okay. The second question is interesting derivative class and integral binding. If this pattern fits, what's known about here? Each category like. to all cell type C makes the arrangement for pole 2 between enhance and promoter. Oh, for pole 2, and let me for pole 2, let me see first for pole 2. Oh, for pole 2, I think it's one is here, one is here, one is here. Yeah, all of them. It's for pole 2. It's like all of them in the 3 different cell lines. They are located in this region, which means that all of them they have like a pattern like in this like this. And how does that fit with 3D proximity between? Oh, that's actually that's a problem, because in this paper we assume that the peak will regulate the nearest genes. That's why we tested the nearest TSS and the near enhancer. But we didn't count any like a 3D proximities in here. I think that's a limitation for this method.
Great. Are there any other questions at this time? All right, so I don't see any other questions in the chat box, and doesn't sound like anyone else is unmuting themselves. So at this point, I would say thank you, Kai. I think we can stop the recording.
Okay, thank you.